A tiny fish moves beneath the opening of a desert cavern in Nevada. This is the Devil's Hole pupfish, and its entire natural habitat exists in just one place: Devil's Hole. So how can one desert pool shape the future of an entire species? The answer begins with a surprising possibility. Devil's Hole may be more than the fish's address. The pool may be part of the biological process that helps make this fish what it is. If so, is keeping a population alive somewhere else enough to save the species? At first, creating backup populations sounds straightforward. If every wild fish lives in one pool, establish additional populations in safer places. Then one catastrophe cannot threaten the entire species. But what happens when the new pool changes how the fish develop? Devil's Hole pupfish, kept in artificial refuges, rapidly developed body shapes that differed from those observed in Devil's Hole. The fish survived, which mattered enormously. Yet their bodies did not develop in exactly the same way. Preserving individual fish does not automatically mean preserving every trait the species develops in its natural habitat. Devil's Hole pupfish have small bodies, relatively large heads and eyes, and no pelvic fins. Their appearance is distinctive, but their evolutionary history is less settled. Scientists still debate how long pupfish have occupied Devil's Hole. That uncertainty limits what we can safely say about their past. One population genomic analysis inferred a more recent and connected evolutionary history among Death Valley pupfishes than the older isolation narrative suggested. But that estimate comes from a scientific model. It is not a settled arrival date. Instead of imagining a creature evolving in complete isolation for a confidently known span of time, we need to keep the timeline unresolved. Since the timeline cannot answer everything, the more useful question concerns development. How can the conditions surrounding a developing fish influence the body that forms? The answer involves developmental plasticity. The term sounds complicated, but the idea is simple. Environmental conditions can influence how an organism develops during its lifetime. Think of genes as biological instructions operating within a changing environment, not as a rigid photograph of the finished animal. The instructions matter, but so do the conditions in which they are carried out. Food, temperature, growth, and hormone-related pathways may interact as a young fish develops. That does not mean one environmental factor created the Devil's Hole pupfish. Nature does not come with a button labeled, "Make unusual fish." How could researchers test part of this idea without placing an extremely endangered population at risk? They studied a close relative, the Amargosa River pupfish. That distinction is essential. The experiments were not conducted on Devil's Hole pupfish, and their results cannot fully explain how every wild Devil's Hole trait arose. Instead, those experiments can show whether certain environmental conditions are capable of shifting pupfish development. Researchers changed food availability, growth conditions, and temperature. Different food rations produced groups with different growth rates. Fish growing under different conditions developed different body proportions, including changes involving the head, eyes, body depth, and pelvic fins. Warmer conditions further reduced pelvic fin development across the experimental groups. Slow-growing fish also showed reduced thyroid hormone levels. What biological pathway might connect these changes? One possible link is thyroid signaling. Hormones are chemical signals that help coordinate processes inside an organism, including development. When researchers used chemicals that inhibited thyroid hormone production, the fish developed a similar cluster of anatomical changes. This does not prove that thyroid signaling is the only cause. It supports thyroid signaling as one possible pathway connecting environmental conditions with body development. Food and temperature can affect growth. Growth and hormone-related pathways may then influence the anatomy that emerges. So what does the evidence support, and where does it stop? The evidence supports interacting effects. It does not identify an exclusive cause, reveal a single master switch, or reconstruct the complete evolutionary origin of the Devil's Hole form. Even with those limits, the results change how we think about refuges. Two pools can contain closely related fish while providing different developmental experiences. If those experiences differ, could the bodies that emerge differ too? The refuge observations suggest they can. But development is only half of the conservation problem. The other half is hidden inside the genome. A genomic study reported severe inbreeding in Devil's Hole pupfish and an elevated burden of potentially harmful genetic variants. Potentially harmful does not mean every variant causes a confirmed problem in every fish. It identifies a concern, not a verdict. Still, what can happen when a population remains extremely small? It can lose genetic options, that may matter across future generations. A head count tells us how many animals are alive today. Does it tell us everything about their genetic health tomorrow? The study reported population bottlenecks of 38 fish in 2007 and 35 fish in 2013. A bottleneck is a sharp reduction in population size. It forces a population's genetic future through an extremely narrow opening. What can happen when so few individuals contribute to later generations? Closely related animals are more likely to reproduce, and potentially harmful variants may become a greater concern. Researchers also identified candidate genetic losses involving genes associated with sperm movement and low oxygen biology. Those associations do not establish their exact effects in wild fish. They are candidates requiring further investigation, not biological verdicts. But alongside severe inbreeding and an elevated burden of potentially harmful variants, they point to a larger problem. Why can't conservation be measured only by whether fish remain alive in a tank? To see the developmental side more clearly, picture the related species experiment as parallel lanes. Juvenile pupfish begin under different food conditions. As their growth rates separate, their bodies develop differently. Their head and eye proportions, body depth, and pelvic fin development can shift. Adding warmer conditions reduces pelvic fin development even further, interfere with thyroid signaling, and a similar cluster of changes appears. This reconstruction illustrates a plausible mechanism in a close relative. It does not recreate the full history of every devil's hole trait. That boundary protects the useful part of the result from being stretched into a claim it cannot support. Now connect the experiment with the refuge observations. Devil's hole pupfish in artificial refuges rapidly developed body shapes that differed from those seen in Devil's Hole. Experiments in the related Amargosa River pupfish show how food availability and growth conditions can shift development. Temperature and thyroid signaling are among the possible influences. What picture emerges when these findings are considered together? Environmental conditions may shape anatomy through growth and hormone-related pathways, but the findings do not identify the exact cause of the refuge differences or prove that any single factor created the species. And here is the counterintuitive twist. Can a refuge reduce one danger while introducing a different conservation challenge? Yes. A refuge can reduce the immediate risk of extinction while creating conditions in which different body forms may develop. That does not make refuges pointless. Keeping fish alive in a refuge can be an essential conservation achievement. But does survival in a new location automatically demonstrate preservation of habitat-shaped traits or genetic health? It does not. A backup population should not automatically be treated as an exact biological duplicate of the natural population. So what should conservation measure instead? At least 4 overlapping layers. First, population presence, whether the fish are alive. Second, habitat condition. Are developmentally relevant environmental features being preserved or reproduced? Third, development. Are the fish continuing to form comparable traits? Fourth, genetic health. What is happening to diversity, inbreeding, and potentially harmful variation? These layers overlap, but they cannot substitute for one another. A positive answer about survival does not settle the other 3. The bigger picture appears only when population, habitat, development, and genetic health are examined together. The Devil's Hole pupfish offers a warning against treating a species like a file that can simply be copied into a new folder. Living organisms develop through interactions between inherited biology and surrounding conditions. Move them, and some interactions may change. Keep the population extremely small, and genetic risks may continue even when every surviving animal receives careful protection. What then should success look like? Success may require multiple measurements across multiple generations. Recognizing differences in refuge populations is not an argument against refuges or intervention. It is an argument for measuring what an intervention preserves, what it changes, and what remains uncertain. When an endangered species is moved, bred, or established in a refuge, which questions should come first?
Begin with the obvious one. Did the intervention help keep animals alive? Then go deeper. Does the new habitat reproduce conditions relevant to development? Do the animals retain body traits comparable to those in the natural population? Is genetic diversity being monitored alongside population size? And which conclusions come from direct evidence about the species rather than experiments on a relative? Each kind of evidence answers a different question. Refuge observations show that Devil's Hole pupfish can develop different body shapes under different conditions. What do the related species experiments add? They reveal possible developmental mechanisms. Genomic findings identify severe inbreeding and an elevated burden of potentially harmful variants. None of these forms of evidence replaces the others. Together, however, they create a more complete picture. What role does uncertainty play in that picture? It can be useful. We do not know exactly when pupfish first occupied Devil's Hole. One genomic analysis inferred a more recent and connected history, but its model-based estimate is not a settled date. We also do not know that thyroid signaling alone explains the wild fish's anatomy. Instead, we know that changes involving growth, temperature, and thyroid signaling shifted development in a close relative. We also do not know the exact effects of every candidate genetic loss in wild fish, even though the genomic warning signs remain important. Keeping those limits visible is not weakness. Scientific caution shows conservationists which conditions deserve attention, which measurements should continue, and which conclusions require stronger evidence. Most importantly, it prevents being alive somewhere from becoming the only definition of success. Now return to that tiny fish beneath the cavern opening. Does its world still look like only a geographic trap? At first it did. One species, one pool, and one dangerously small habitat. Now the cavern represents something more. It contains conditions that may influence development. It holds a population whose genetic future has passed through severe bottlenecks. And it contains an evolutionary history that remains partly unresolved. Devil's Hole is not merely where this fish lives. It may be part of the developmental context that conservation must learn to protect, while the population's genetic health requires its own careful monitoring. So how can one desert pool shape the future of an entire species? It may influence not only where the fish survives, but also the conditions in which its body develops and its genetic future unfolds. Saving the fish may ultimately require understanding the water around it.